It is. Okay, so I'm going to talk about a project that we recently completed. So this sort of ended in the last couple months, but I'll give you some results on PFAS in fish in the Great Lakes. The people on the lower left there actually did all the work. And also some funding was provided as well on a concurrent project with the U.S. Geological Survey, and collaborated with U.S. Fish and Wildlife Service, and Notre Dame, through the Environmental Change Initiative, provided some funding as well. So our objectives, generally speaking, really were to kind of get a snapshot of PFAS. This, I think most people know what PFAS is. Yes. Okay. Toxic, forever chemicals. Okay. So I won't explain those too much. To get a snapshot of PFAS in Lake Michigan sportfish and also in prey fish as well. So the second objective was to try to evaluate these dietary routes for PFAS as they penetrate into sportfish in Lake Michigan and those that migrate upriver as well. Next slide. So I'll give you the key findings right now so you're not in suspense. Every fish that we sampled out of Lake Michigan had detectable levels of PFAS in their tissues, in their muscle tissue, and in their other organs as well. PFAS, this sort of legacy chemical that's been phased out but is still very persistent, was most frequently detected and was highest in concentration in all the fish that we tested from Lake Michigan. The good news is that PFAS concentrations in Lake Michigan fishes were actually lower than some of the other Great Lakes, such as Lake Ontario. Still there, but at least they were lower than some of these others, but still present. And also, finally, PFAS accumulate differentially among the organs of fish, and especially the embryos, which I'll get to in a moment. Next slide. So here are some data. These are this paper, these data right here are in a paper that's in revision right now. It's Stoughton, Science of the Total Environment, led by a great postdoc, Daniela Miranda, that you heard from about a year ago at this same meeting. So on the left are the concentrations in predator fish, mostly salmonids, well, all salmonids actually, and then prey fish up above. So the lettering's a little small, but you can get the idea. To the right are relative abundances of different PFAS compounds. There's a whole slurry of them, thousands of PFAS out in the environment. So that's relative concentration on the right. So notice that predator fish in general have higher concentrations than prey fish, so some biomagnification occurring. The blue is PFAS, so the dominant compound, but there are many others there as well. Interestingly, sculpin were loaded with PFAS. So that's a very interesting one. We're trying to decipher that one as well and talk to other experts as to why sculpin are so loaded, bottom dwelling fish, et cetera. Next slide, please. So this paper right here, we also took a look at where PFAS are going within the bodies of salmon. And this paper right here exploded in late 2022, was published in Environmental Science and Technology Letters by a great PhD student, Whitney Conard. And it's gotten over 500 hits on the American Chemical Society website alone. So it's many people are aware of this. So just Google PFAS and fish eggs, and this is the first hit. The bottom line is that female salmon are offloading their PFAS to their embryos. Okay, very sad. But this PFAS is penetrating into the eggs of salmon. And of course, salmon run upstream, sometime to a hatchery, sometime into a naturalized stream. And then they're releasing their eggs. And then other resident fish, such as that brook trout on the image on the lower left, are eating these eggs, are gorging on them. So they're getting their load of PFAS as well from these eggs. And of course, people are angling for these fish and presumably eating them as well. So it's very interesting. In the lake, PFAS concentrations are the same in males and females of salmonids. And then when they go upriver, such as coho and chinook and others, females are offloading their burden to their eggs. Next slide. Next steps in this project, very grateful to Sea Grant because it's allowed us to leverage some other funding currently from the Great Lakes Fishery Trust to look at some of these tissue differences among fish and also different trophic levels. Biotransport by the Epley Foundation in the bodies of salmon. We have an upcoming project with the Indiana Department of Natural Resources through the Lake Michigan Coastal Program to look at the distribution of PFAS in tributaries in southern Lake Michigan. And the Illinois-Indiana Sea Grant will be seeing a proposal in about 10 days to sort of build on that project, working on water and sediment to add the biota in these tributaries to see where PFAS are going. Next animation, real quickly. Some products from this funding. Again, there were concurrent projects with Sea Grant and a related but different project with the USGS. This produced 3 PhD theses and 1 in progress. 2 published papers already, a submitted paper, the one you saw on the concentrations in fish, several manuscripts, a lot of conference presentations, web interviews, and an NPR broadcast. Next. But what I'm most proud of is that this has trained a postdoctorate, currently a research assistant professor, Daniela Miranda, 8 graduate students, including your own Catherine O'Reilly, and also 10 undergraduates have worked on this, many of which are women or underrepresented minorities. So thank you, Illinois-Indiana Sea Grant.